This is an OCR A-level biology video on membrane-bound organelles, organelles without membranes, and looking at those cells, and then the making and secretion of a protein, as well as comparing eukaryotes and prokaryotes. This is following on from my previous video on Unit 2.1 on microscopy, but most of you are coming from that video, so let's just get started. Here, that's a little sneak preview of last video. So, membrane-bound organelles. We're going to look at each one individually. First of all, nucleus. So a nucleus has a nucleolus, a nuclear envelope, and chromatin. They're the three main things that I'm going to speak to you about here. The nucleolus has no membrane. It contains RNA, and it's where ribosomes are made. Our nuclear envelope is a double membrane which separates the contents of the nucleus from the cytoplasm. It contains pores to enable large substances such as mRNA and hormones mRNA and hormones in. When the two membranes fuse together, it allows dissolved ribosomes to pass through. Okay. Then we've got this chromatin, and chromatin is genetic material consisting of DNA wound around histone proteins. When the cell is about to divide, chromatin condenses and coils tightly into chromosomes, and chromosomes contains the organism's genes. Okay, so the functions of the nucleus are to control the centre of the cell, store organism's genome, transmit genetic information, and provide instructions for protein synthesis. Then we've got the rough endoplasmic reticulum, or RER, and within our RER we've got cisternae, which are fluid-filled cavities. Okay, it's also got ribosomes. That's why it's rough. It's got ribosomes coating it, and so that's the two basic things about it. So cisternae is a fluid-filled cavity, continuous with the nuclear membrane, and the RER's got an intracellular transport system role. Okay, so basically move proteins between the nucleus and the Golgi apparatus, which we'll see later. The ribosomes coat our rough endoplasmic reticulum, and they also assemble amino acids into proteins, take it to the cisternae, which then takes it to the Golgi apparatus. Okay, that's the basic process. So now let's look at the smooth endoplasmic reticulum, which does a very similar role. It does not have ribosomes on its surface, however. It also has this cisternae system. Okay, so it's got fluid-filled cavities that are continuous with the nuclear membrane. It has a role of absorbing lipids from the gut, and it contains enzymes that catalyse reactions involved in lipid metabolism. So it undergoes synthesis of cholesterol, synthesis of lipids and phospholipids needed by the cell, and synthesis of steroid hormones. So next we're going to look at the Golgi apparatus, and this is another organelle involved in protein transport. So the Golgi apparatus modifies proteins, so it can make glycoproteins by adding sugar molecules, and it can add lipid molecules to make lipoproteins and folding them into their 3D shape, so it is responsible for making the protein's tertiary structure essentially. The proteins are packaged into vesicles that are pinched off and then stored in the cell or moved to the plasma membrane, either to be incorporated within the membrane or to be exported outside the cell. They contain a stack of membrane-bound flattened sacs, and they've just got roles in transporting vesicles around. Okay. One type of vesicle is a lysosome, and they are small bags formed in the Golgi apparatus, each surrounded by a membrane. They contain hydrolytic enzymes. In phagocytosis, we see lysosomes play a big role in digesting invading bacteria, invading fungi, etc. Okay, so they can digest cells, and we also see that happen in apoptosis. So when cells, programmed cell death, cells die. They basically consume themselves, and that is done by lysosomes and hydrolytic enzymes. Okay. Then we've got mitochondria, which has a matrix and cristae, and it's got a double membrane. So it's got this outer membrane, then we've got the cristae, then we've got the matrix. The matrix is the fluid filled area. The cristae is where we see oxidative phosphorylation take place in respiration, which forms ATP. So that's the major role of the mitochondria, is to form ATP. It's a 2 to 5 micrometer spherical rod, and it contains mitochondrial DNA, mDNA, which means the mitochondria are self-replicating. So chloroplasts are only found in plants. They've got a double membrane as well, with a stroma and grana, much similar to the mitochondria. The stroma is the fluid-filled matrix, which holds the light-independent stage of photosynthesis, such as the Calvin cycle. The grana have flattened stacks of thylakoids, which contain chlorophyll, and each stack is called a granum. The fluid-filled matrix is the stroma. They're abundant in the palisade mesophyll layer, which is just below the upper epidermis in the plant structure. And they're 4 to 10 micrometers long, so they're pretty much twice the size of mitochondria. Okay, so photosynthesis in chloroplasts, respiration in mitochondria. Then we've got the vacuole, which is one of our last membrane-bound organelles. It's got this membrane called a tonoplast, which contains fluid, and it fills with water and solutes, maintaining cell stability, because when it is full it pushes against the cell wall, making it turgid. So a huge role in turgidity in the vacuole, just to support the plant and to prevent placid tissue or flaccid tissue forming. Finally, which I have to squeeze in the bottom here, is cilia and undilopodia, which cilia you will recognise from epithelial cells. Undilopodia are found in spermatozoon. They're formed from centrioles. They contain microtubules, protrusions from the cell surrounded by the cell surface membrane, which is basically what they are. They contain receptors and allow the cell to detect signals about its immediate environment. So the cilia have a role in beating and moving bands of mucus within our epithelial tissue, within the bronchi, trachea. Right, so that was your membrane-bound organelles. I've really gone through them quite quickly. One thing that was being written here while I was talking, I put it here because there was a huge white space, which was really annoying. There's another one there, but sadly I cannot fill with any words. One thing I wrote here in blue, you may have been annoyed because it was being written while I was not even talking about it, and that is ribosomes. So ribosomes, we saw them in the rough endoplasmic reticulum. They are also an organelle. However, they do not have a membrane. They do not really belong on this page. That's why I've highlighted them in blue, and I'm going to speak about them now, just before I look at all the other organelles without membranes. So ribosomes, they're found on the exterior of the RER for synthesis of proteins, but they can also be found free in cytoplasm of the cell to assemble proteins that will be used within the cell. And within them they contain ribosomal RNA. They have a 20 nanometer diameter and a spherical sort of shape, and they do not have a membrane. Let's remember they are made within the nucleus. They pass through the envelope, and to pass through that envelope, that double membrane, the double membrane fuses together, both membranes fuse together to allow ribosomes to passage through in sort of two parts, two subunits. Okay, so now let's move on to non-membrane bound organelles, so just organelles without membranes, ribosomes being the first one, and that's the first thing I write here on the left hand side about ribosomes. We've already looked at them, but the next one is centrioles. So centrioles, you'll recognise them from mitosis and meiosis. So centrioles are two bundles of microtubules at right angles to each other, made from tubulin protein subunits. Before cells divide, threads of tubulin form spindles. Chromosomes join to the middle part of the spindle, and motor proteins walk along tubulin threads, pulling chromosomes to the opposite ends of the cell by their centromeres. They're also involved in the formation of cilia and undilopodia, so they multiply and line up beneath the cell surface membrane and then sprout outwards from each centriole, forming a cilia or undilopodia. Okay, they're absent from cells of higher plants but might be present in unicellular green algae. So that's centrioles, an organelle with outer membrane. So next we've got cytoskeleton, and the cytoskeleton has many different roles within a cell. The main one just being to give the cell strength, stability, and allow movement. Okay, so movement of substances through cells. It also anchors the nucleus within the cell, and it forms a spindle for cell division. So it's very similar to, got a similar role to centrioles. It's involved in microtubules, straight cylindrical microtubules made of tubulin again, 18 to 30 nanometers in diameter. It's basically what it's formed of. It's got intermediate filaments and rod-like filaments made of subunits of protein actin, which are 7 nanometers in diameter. Cytoskeletal motor proteins can be myosins, kinesins, or dynesins, and they drag these motor proteins drag organelles or substances such as chloroplasts through the cell. I believe it's an active process, so it involves ATP energy, and they just basically involved in the movement of cells. So cytoskeleton, cell signalling roles, movement of substances within a cell, cell stability and strength, anchoring that nucleus, forming that spindle. Okay, huge roles within the cell, really essential but often unnoticed or unrecognised. So then we've got this cell wall, which is our final organelle without a membrane, and it's for strength, support and shape. It is fully permeable, but its permeability can be altered by addition of different ions. Then we've got this final point here, that plants, fungi, Prokaryotes all have cell walls. Okay, bundles of cellulose fibres. It's absent from animal cells. So cell walls can be made of cellulose or chitin, which is in fungi. Prokaryotes are that funny group, aren't they, which have animal and plant features, and some have cell walls, some don't. Alright, it's all variable, and Prokaryotes is a very hard group to understand. What we've got here is two pictures. I'm going to put a third picture on after. So here we've got a animal cell. Here we see the nucleus, and it's got this double membrane around the outside. The nuclear envelope contains DNA within here, bound around histone proteins. We've got Golgi apparatus drawn here, mitochondria here, a single mitochondrion, endoplasmic reticulum, which would actually be attached to our nucleus. We can't actually tell which endoplasmic reticulum that is there, because ribosomes are so small you can't pick them up on light microscopes. They're, I believe, the 20 nanometers. That might be completely wrong. The ribosomes are extremely small, organelles, 20 nanometers in diameter, and they can't be picked up by these microscopes. So that is what you might have to deal with in the exam. Don't expect huge clarity, and sometimes it's quite hard to depict each one, but just use your head and try and cancel out which ones it can't be. Okay. Here we're looking at basically the protein, the ones involved in forming proteins. We've got the nucleus, it sends these proteins out. I'll speak to you about the formation of proteins in the next slide. the rough endoplasmic reticulum, and our Golgi apparatus, which is free in the cytoplasm. Okay, and vesicles are transported between the three. And the difference between rough and smooth is rough has the ribosomes, smooth does not. Okay. Our final picture is this one, which is much more detail and much less likely to see something like this in an exam. It's sort of cartoon-like because you can't really picture a cell being like this. So you've got the large nucleus. Around it we've got this rough endoplasmic reticulum which surrounds it, the smooth endoplasmic reticulum. We've got a lysosome here, centriole, Golgi apparatus attached to our rough endoplasmic reticulum, ribosomes, mitochondria. Okay, you can look at it for yourself. If you want to pause it and have a little study of these diagrams, feel free to do so. But now I'm going to move on to proteins and how they're formed. Okay, so making and secreting proteins. So the gene that has coded instructions for a protein, such as insulin, is housed on chromatin in the nucleus and transcribed into a length of RNA. So the genes with the instructions for the protein is transcribed into a length of mRNA, messenger RNA. Many copies of this mRNA are made, and then they pass out of the pores in the nuclear envelope through ribosomes. So they use ribosome. They attach to ribosomes, which then translate the instructions on the mRNA, producing a polypeptide chain. And you can look in transcription and translation on the videos. I've done that in detail. I'm not going to speak to you about how the process happens. Just remember there's transcription, then translation, then we produce this polypeptide chain. And this polypeptide then passes into the cystine, which is our fluid-filled matrix of the RER, rough endoplasmic reticulum, along hollow sacs. The vesicles within the with polypeptide inside are pinched off from the rough endoplasmic reticulum and pass via microtubules and motor proteins to the Golgi apparatus. The vesicles fuse with the Golgi apparatus, which the molecules then pass to the plasma membrane. So vesicles and plasma membrane fuse, and the protein can be released out of the plasma membrane, or we can just release the proteins from in vesicles through the Golgi apparatus, which can modify some proteins, and they just then stay in the cell cytoplasm. So they're stored in the cytoplasm, or they can fuse with cell membranes and be released. And the movement of them through cell membranes will therefore be through our cytoskeleton. Finally, we're going to compare eukaryotic cells to prokaryotic cells. So what I'm going to do is just list the thing here, so like plasma membrane, cytoplasm, and then compare what that structure or how it happens in eukaryotes and prokaryotes. So I'm going to actually write this in as I'm talking. Then to summarise it, we'll look at this diagram showing the difference between the two at the end. So, plasma membrane. Both prokaryotes and eukaryotes have a plasma membrane. What I was going to do is do one in blue and one in red, see how that goes, but it might take a longer time, so I'm just going to do. No, it's going to take way too long to keep changing the colour. So what I'm going to do is just use a neutral colour. We'll use grey. And eukaryotes and prokaryotes both have plasma membranes. They also both have cytoplasm, okay? And they both have ribosomes. So eukaryotes have much larger not much larger ribosomes than prokaryotes. Different sources tell you different things, but eukaryotes have 80 S, whereas prokaryotes have 70 S, which is the unit for size. The important thing here is, let's just remember, ribosomes in prokaryotes are smaller than eukaryotes. Then we can look at DNA. So in eukaryotes, by the way, both eukaryotes and prokaryotes have DNA, so let's give them a tick. But in prokaryotes, the DNA is not wound around histone proteins. It is free. So we'll put free DNA here. But in eukaryotes, it is wound around histone proteins. I'll just put histone proteins. This is why I usually don't write during the presentation, because I find it hard to multitask. So prokaryotes is free, and usually they're in a plasmid, which contains their genetic material, which is like circular things. Okay. So RNA, both prokaryotes and eukaryotes have RNA. Prokaryotes probably rely on RNA more than eukaryotes do, but they both have it. Size, eukaryotes are much larger than prokaryotes in general, so large and small. So prokaryotes are smaller than our eukaryotic cells. Then we have centrioles. So eukaryotes have centrioles, but prokaryotes don't. And let's remember they're a non-membrane bound organelle. Membrane bound organelles, eukaryotes do, prokaryotes do not. This is not a good cross. Prokaryotes do not have any membrane bound organelles. That includes the nucleus. Let's remember they have free DNA, or DNA in plasmids. They have no membrane bound organelles. They have different organelles to respire. Do not have any membrane bound organelles. No Golgi apparatus, no SER, no RER. Okay. Cell wall. Yes, they both have a cell wall, but in eukaryotes it's cellulose or chitin. Cellulose or chitin. Not all eukaryotes have a cell wall, by the way, but prokaryotes can also have a cell wall, but it's made of peptidoglycan. So that's what it's made out of, and their method of replication is different. So in prokaryotic cells they undergo binary fission, whereas in eukaryotic cells they undergo mitosis. And you do not have to know how binary fission works. We do not study it in our specification. So they're the differences between eukaryotic and prokaryotic cells. Just learn those differences. There are obviously more, like prokaryotic cells have flagella, which are like long whip-like projections enabling them to move. They also have pili, which are smaller hair-like projections that allow a bacteria to adhere to host cells or to each other and allow the passage of a plasmid between one cell and another. So they've got flagella and pili, which are not found in eukaryotic cells. Finally, let's just compare the two. So first of all, we see there's a huge difference in size. Eukaryotic cell on the left, much larger, much smaller. So here we've got a flagella there. Nucleus, SER, RER, Golgi apparatus, mitochondria, all those membrane-bound organelles are in our eukaryotes. They're not present here. We've got DNA within here wound around histone proteins as chromatin, but we've got free DNA in plasmids here. Smaller ribosomes, larger ribosomes, they're the main difference you're going to sort of pick up on. The rest of them are out here. That is a prokaryotic cell, that is a eukaryotic cell. That is the end of Unit 2.1. Just one more video to come from me on Unit 2 now, and that's going to be on enzymes, coming very soon. Thank you for watching.